안녕하세요. 오늘 소개해 주셔서 감사드리고 오늘 웨비나에 참여해 주셔서 감사합니다. 저는 오레곤 보건과학대학교에서 현재 포닥 연구를 진행 중인 허동은이라고 합니다. 오늘 발표는 사실 영어로 준비를 해서 영어로 발표드리겠습니다. Uh, Today, I'm excited to present to you on my work from my PhD lab, where I looked at the transcriptional changes in oligodendrocyte precursor cells across aging in the lab of Dwight Burgles. This work was recently published in Nature Aging, and I'm really excited to share you the results from the experiments. So before we start, I have to introduce you one of my favorite cell types in the brain, which are oligodendrocytes. Oligodendrocytes are the myelinating glial cells of the central nervous system, and they extend their cytoplasmic processes and wrap around neuronal axons, forming these concentric wrappings along the axon. Upon compaction and extrusion of the cytoplasmic cytosol, you can actually see that these form very compact structure, also known as myelin sheets. And these myelin sheets are most famously known to facilitate saltatory conduction along neuronal axons. However, more recently, it's also been shown that through myelin sheets, oligodendrocytes can provide metabolic support and regulate potassium homeostasis. And interestingly, oligodendrocytes can somehow respond to neuronal activity and dynamically modulate different features of myelination to facilitate learning and memory. So oligodendrocytes, which you can see here, are actually generated from a pool of self-renewing progenitors called oligodendrocyte precursor cells. I'll be referring to them as OPCs throughout the talk, but what I mean by OPC is oligodendrocyte precursor cell. These oligodendrocyte precursor cells, like I mentioned, can proliferate and self-renew, and they're also quite dynamic. So here is a video of in vivo imaging data from mouse cortex from Dwight Burgles' lab. And what you can see is that these OPCs are highly dynamic, where they dynamically extend and retract their processes. And you can see that these one OPC can undergo proliferation to generate two OPCs. In addition to undergoing proliferation, OPCs can differentiate into pre-myelinating oligodendrocyte, and this change requires dramatic change in morphology, transcription, and metabolism to sort of prepare for the cell to make tons and tons of membrane myelin structures when they become mature oligodendrocytes. And some of these pre-myelinating oligodendrocytes continue to mature and become myelinating oligodendrocytes in the brain. So if you were to look at the whole mouse brain, so this is a sagittal section of a young adult mouse brain, and you might imagine that OPCs, being the progenitors, might not be present in the adult brain and might be only transiently present in developing brain. However, even in the adult, here stained by marker NG2, OPCs are present throughout the brain. And if you were to look at closely, you can see that OPCs tile with one another and that they occupy non-overlapping territories throughout the brain. However, even though OPCs are present in the brain throughout life, the actual rate of generating new oligodendrocytes decline with aging. So here on the left, it's like a summary of the data from Bill Richardson's lab. So here in green, what I'm representing here is the number of total oligodendrocytes in the brain across aging. So you can see that earlier in development, new oligodendrocytes can form very rapidly. However, as the brain ages, the rate of new oligodendrocyte production dramatically declines. And that is paired by proliferation of OPCs. So here in purple, we're looking at now the OPCs that are undergoing proliferation. And you can see that early in development, there is a high degree of proliferation of OPCs, and that also dramatically declines over the course of aging. And this can also be shown in a different way, where if you were to look at the white matter volume across human age, you can see that earlier in development, there is a massive increase in the volume of myelin and volume of corpus callosum in humans. However, this increase in new myelin produced by oligodendrocytes declines with aging, and then actually we lose myelin over the course of late aging brain. So now the question is like, why is there this sudden decrease in new oligodendrocytes and new myelin formation when we have a constant pool of progenitors throughout life? One of the reasons could be that oligodendrocyte lineage cells, including OPCs, can become senescent with aging as well as in neurodegenerative diseases. So here I have examples of multiple sclerosis as well as an Alzheimer's disease where they can stain for markers of senescence or P16 or P21 and show that oligodendrocyte lineage cells upregulate these markers of senescence. Now the question is, then what are the cell intrinsic changes in OPCs that happen in the aging brain? So we know that the aging brain undergoes a ton of neuroinflammatory changes, mostly driven by reactive microglia and astrocytes releasing pro-inflammatory cytokines. And now in that environment, how do OPCs respond and how does that change their cellular behavior? In order to ask this big question of how does aging change OPC transcriptome and dynamics, we actually started out by making a new transgenic mouse line. So we made a new OPC reporter mouse line using this gene called matrillin-4. So here on the top is a bulk RNA sequencing from the Barris lab, where on the x-axis, you're looking at different population of neural cells, astrocytes, neurons, oligolineage, endothelia, and microglia. And you can see that this gene matrillin-4, MATN4, is highly enriched in OPCs and newly formed oligodendrocytes, and pretty much absent in other neural cell types in the brain. This is, and on the bottom is a single nuclear RNA sequencing data from Greenberg lab at Harvard. And here you can also see that matrillin-4, this gene that we're interested in, is enriched in OPCs and newly formed oligodendrocytes and pretty much absent in any other neural cell types in the brain. And this expression pattern is similar to known markers of oligodendrocyte precursor cells, CSPG4, also known as NG2, as well as PDGF receptor alpha. So after figuring out that matrillin-4 is a good gene to target to make a new reporter mouse line, what we did was to insert a fluorescent protein, in our case, membrane-targeted EGFP, into the first coding exon of matrillin-4 gene. Here in the first coding of exon of matrillin-4, I've knocked in membrane anchor EGFP, WPRE, and poly-A using CRISPR-Cas9. And if you were to look at the mouse brain of this animal, you can see that all of the EGFP positive cells co-localize with no markers of OPCs, PDGF alpha, and NG2. If you were to zoom in, you can see that EGFP positive cells are double positive for PDGF receptor alpha and NG2. But the EGFP signal is actually absent in PDGF receptor alpha only perivascular fibroblasts, further showing the specificity of this reporter mouse line to oligodendrocyte precursor cells. So once we had this mouse line, we were excited to perform single-cell RNA sequencing. And the reason we decided to perform single-cell RNA sequencing over other methods of profiling is because OPCs are inherently very heterogeneous in their cellular behavior. At a given time point, most of the cells are going to be quiescent, but some of the cells are going to be undergoing cellular proliferation, and some of them will be undergoing differentiation to make new oligodendrocytes. So instead of taking the whole tissue or just taking a cell type and performing a bulk RNA sequencing, which could dilute the effects of different OPC behavior, we decided to perform single cell RNA sequencing so that we might be able to resolve different states of OPCs in different clusters of cells bioinformatically. So using the matrillin-4 MEGFP CRISPR knock-in mouse line that I just described before, we took both males and females at P30, P180, P360, and P720 to sort of get a broad understanding of how OPCs change across the whole lifespan of the mouse. We took their brains, isolated their cortices, and performed Miltenyi neural tissue dissociations to acquire single cell suspension. Took those population of cells and performed FACS isolation of GAP positive OPCs. And this particular step was actually very important because OPCs only comprise of only like 2 to 5% of the whole neural cells in the brain, which meant that I really needed to enrich for the population of my interest, OPCs, in order for me to get a high resolution OPC data set at the end. So once I had the enriched isolated population of OPCs, I took those cells, generated single cell libraries using the 10x Chromium system and sent it for sequencing. So at the end of this journey, I ended up with 38,807 cortical OPCs represented from 4 different ages that I talked about, P30, P180, P360, and P720. And then I was able to resolve different subtypes of OPCs. And these different subtypes actually represent different states of oligodendrocyte precursor cells. So again, the biggest population of the subtypes represented in my data are the quiescent OPCs. I do have a very nice population of proliferating OPCs, as well as the OPCs that are undergoing differentiation. So now the question was, how do these different subtypes of OPCs change with aging? So here on the left this is the same graph as before, just to show you what my data looks like on the UMAP plot. In the middle, you see bar graphs of different OPC subtypes in 4 different age groups. So here you can visually see that the blue bar is increasing. So those blue bars are quiescent OPCs. And you can see that over the course of aging, more cells are becoming quiescent and less dynamic. With the increase in quiescent OPC population, you can also visually see that the differentiating population is massively declining, just as we expected. And you can also see that some cycling populations are decreasing in proportion. Here's a different way to look at the same exact data, here now separating them into different subtypes of OPCs. You can see that over the course of aging, so blue to purple, you can see that cycling OPC3, transitioning OPCs, and differentiating OPCs are the populations that are undergoing significant change with aging. So with that, it was basically a good proof of principle that using single-cell RNA sequencing, we can reveal the OPC dynamics that we've known before, that aging induces quiescence in these population of cells, and then less cells are going to be differentiating and less cells are going to be proliferating. But here, what was interesting about my data was that I had different subtypes of cycling OPCs. So instead of a single population of small proliferating cells, now I have this large population of OPCs, cycling OPCs that are somehow bioinformatically distinct. What is the difference between all of the subtypes? So what we found using some bioinformatic approaches, here I use tricycle. I found that the different cycling OPC populations actually occupy different stages along the cell cycle. So here is like the typical cell cycle stages. So G0, G1 phase over to S phase to G2 phase to the mitotic M phase. And you can see that in the population of cycling OPCs that I was able to recover, I have a large population of cycling OPCs, which are predicted to be in that G1, G0 phase in cell cycle. And you can see that this dark yellow population, cycling OPC2, are predicted to be occupying beginning of the G2 M stage of cell cycle. And you can see that these final green OPCs, the cycling OPC3, are predicted to be actively undergoing mitosis. And with that, what you can also do is now look at what are the genes that are differentially expressed across this transition. And one of the things that I looked at was expression of different cell cycle genes as a proof of principle. And I was able to show that all across the pseudotime of this cycling OPC progression from cycling OPC1 to 2 to 3, you can see that different transcription factors and cell cycle related genes are differentially expressed along that time course. With that, I want to sort of like wrap up talking about cycling OPCs. I have more data in the paper, but I sort of want to move into talking about this population of cells, which are the cells that are actively undergoing differentiation. As I mentioned before, this change going from OPC to pre-myelin oligodendrocyte is like a huge change, both morphologically and transcriptionally. So I really wanted to understand what are the regulators that govern OPCs to go from quiescence to differentiation. So I took this group of cells and then reclustered them to get a better resolution of what's happening at this particular stage. So here is the same plot, just isolated for the late stage of quiescent OPCs as well as the transitioning and differentiating OPCs. And you can see that there is a difference in the expression of oligodendrocyte lineage genes across this time course, going from quiescence to differentiation. So this fine resolution of how transcription factors and genes change along this time course has not been fully studied before. So here you can see that HES5 ID2 ASCL1 is highly expressed in this transition quiescent OPC and transitioning OPC state. And then they quickly shut off as OPCs start to actually differentiate. And that is coincided by massive increase in MERF, which is an essential transcription factor for oligodendrocyte differentiation and survival. And here OLIG2 and SOX10, which are pan-oligolineage marker, and they sort of stay flat throughout the whole transformation. So we wanted to understand more about what these transitioning OPC populations are and what are the different markers that could potentially hint at their role in this transition. So here we found several differentially expressed genes, three of which are GJC3, KCNA1, and MAF. And you can see that these three genes are selectively downregulated in this transitioning OPC population. And what was interesting to us was that this downregulated genes were actually increased over the course of aging, if you were to look at this population across aging, suggesting that perhaps downregulation of these factors could be important for dictating whether OPCs can undergo successful differentiation or not, given that the success of OPC differentiation is much higher in the younger brains compared to the older brain. Another interesting gene we found is TPT1, and this is in contrast upregulated ever so slightly in the transitioning OPC population. And what was also interesting was that this gene was now being downregulated over the course of aging, also suggesting that perhaps this is a positive regulator of OPC differentiation that gets downregulated over the course of aging. That was the story on the differentiation of OPCs. And now I want to turn our focus into the biggest population of cells we have in the data set, which are the quiescent OPCs. So I wanted to understand what are the aging-associated changes that occur in this large population of OPCs, which we call quiescent. So at a first glance, what you can notice is that there is this dramatic separation between the OPCs isolated from P30 versus all other age groups. Again, different positioning on the UMAP doesn't necessarily dictate difference in their transcriptome, but it was hinting at us that there is some large transcriptomic change that is separating out the young OPCs from P30 brains with all other OPCs from the older brains. So we then asked, what are the changes that occur in this aged or older mouse brain OPCs that separate them from the young OPCs? And what we found is that we were able to find a group of genes that are highly expressed by the older OPCs compared to the young OPCs. And one of the genes that was really highly differentially expressed was this gene called C4b. And C4b is a component of the complement system, and it has previously been shown to be dramatically upregulated in astrocytes in an aged brain. It was also shown to be upregulated by potentially aged oligodendrocytes and OPCs in the hippocampus. So we were happy to see that we were able to identify this molecule, which seems to report the aging of the brain by all different types of glial cells. So taking all the different groups of all the different genes that are differentially expressed by the aged brain, we then performed a QIAGEN attenuated pathway analysis, or IPA, to identify potential upstream regulators of the transcriptomic change. So here in orange are the pathways that are predicted to be activated in OPCs with aging based on the transcriptomic data. And in blue are the pathways that are predicted to be inactivated with aging. So perhaps most unsurprisingly, we found that inflammatory and immune pathways are predicted to be activated with aging in OPCs. And this makes sense because we know that as I mentioned in the intro, that the aging brain experiences a lot of pro-inflammatory neuroimmune changes driven by different glial cells. Another potentially unsurprising finding was that we saw inactivation of cell growth and survival pathways, and this is mediated by MYC and CROSS. And previously, work from Robin Franklin's lab have shown that MYC indeed is inactivated in aged OPCs, and reactivation of MYC can rejuvenate the ability of OPC to become more differentiated. So two of the more interesting things that I saw from the upstream regulator analysis was HIF-1 alpha and beta-catenin and Wnt signaling pathways. So we focused on HIF-1 alpha and Wnt to see if we can pharmacologically manipulate these pathways and see if we can change the behavior of OPCs in vitro. So first of all, we validated that HIF-1 alpha at the transcriptional level is indeed increased with aging in OPCs. So here, the color of the dots represent the mean expression level of OPCs across time. And we also performed fluorescence in situ in mouse cortex to validate that indeed HIF-1 alpha is increased in expression in OPCs in the mouse brain when the mouse is aged to about a year old. We can also look at the protein of HIF-1 alpha to see if HIF-1 alpha is upregulated not only at the mRNA level, but also at the protein level. So we used a HIF-1 alpha protein antibody to stain the mouse cortex with NG2 as the marker for OPCs. And we can see that compared to P30, P70 brain has a lot more HIF-1 alpha immunoreactivity throughout the brain, and some of them actually co-localize with NG2 marker. If you were to zoom in, it's a little bit more clear that P720, the 2-year-old OPCs, have a lot higher expression of HIF-1 alpha compared to the OPCs from P30 young brain. And if you quantify the percentages of OPCs that have HIF-1 alpha immunoreactivity, that massively increases with aging. And you can also recapitulate this in vitro. So if you were to culture OPCs from young versus aged mouse brain and immunostain for SOX10 marker for oligolineage, as well as HIF-1 alpha, you can see that compared to the young OPCs isolated from the young mouse brain, OPCs isolated from the aged mouse brain have a massively increased immunoreactivity to HIF-1 alpha. So once confirming that HIF-1 alpha does seem to be activated in OPCs with aging, we perform pharmacological inhibition to see whether we can restore the differentiation effect potential of OPCs in vitro. So again, we isolated OPCs from young adult and aged adult mouse brains, generated primary OPC cultures, and used this drug called CAY10585 to inhibit HIF-1 alpha signaling globally. And what we saw is that if you were to quantify the MBP positive myelinating oligodendrocytes in culture, comparing the control to the drug treated, HIF-1 alpha inhibition can increase the differentiation of OPCs in both young and aged OPCs, suggesting that HIF-1 alpha activation could be detrimental to OPC differentiation, and inhibition of that pathway or that activation can increase the differentiation potential of OPCs. We can also do a similar experiment targeting the Wnt signaling pathway using 2 different drugs to target different aspects of Wnt signaling, IWP2 to globally block Wnt signaling, and XAV939 to selectively block canonical Wnt signaling pathway. And here, in the aged OPCs or OPCs isolated from the aged brain, both drugs were able to dramatically increase OPC differentiation. However, in the young OPCs, these drugs had minimal effect or smaller effect, suggesting that aging-induced Wnt activation can be selectively blocked by Wnt inhibition, and that in young OPCs where Wnt signaling has not yet been fully activated, inhibition of the signal does not have the same effect as the drugs do in the aged brain. So to summarize what I've shown you today, that matrolin-4 GFP mouse line allows us to perform in vivo labeling and visualization of OPCs, and allows you to selectively enrich for OPCs for further biochemical, transcriptional, any analyses. I've generated a large single-cell RNA-seq data set of OPCs, and this allows us to now look at transient OPC subtypes that were minimally represented in previous data sets. So cycling, transitioning, and differentiating OPCs. These are all transient states that OPCs can occupy. And now with my data set, we can actually look at what are the transcriptional markers and pathways that are rich in these transient population of cells. I've shown you that quiescent OPCs seem to undergo a large aging-associated transcriptional changes. I've seen, based on the engineered pathway analysis, upstream regulator analysis, I've shown you that quiescent OPCs seem to have increased immune and inflammatory pathway activation, increased HIF-1 alpha and Wnt signaling activation, whereas the cell growth and survival pathway seems to be inactivated in quiescent OPCs with aging. And importantly, we can pharmacologically inhibit HIF-1 alpha and beta-catenin signaling pathway to restore the differentiation capacity of OPCs in vitro, which then allows us to think about therapeutics. How can we increase the ability of OPCs to make new oligodendrocytes and new myelin in injury, disease, and aging? And now the aging OPC data set that I present to you today is publicly available, and it can be easily accessed online. And I'll show you a little bit of how to navigate the website, because I really want a lot of people to sort of like query the data and find cool things and follow up on interesting pathways and genes. So here, if you were to click this link, it will take you to this website. So I've hosted the data set on Chan Zuckerberg Initiative CZI Cell by Gene platform. So here you can not only look at the data, but also download the data set so that you can analyze it yourself. But if you didn't want to download the raw data and just wanted to play with the data, you can click on Explore. And what it will take you is the single-cell RNA-seq data set that I've presented to you today. Here you can colorize them in different metrics. So here I'm colorizing them by their identity or subtype, where the quiescent OPCs are here, cycling OPCs, differentiating OPCs. What you can also do is now you can search for your gene of interest, which may be here an example of BCAS1, which is known to be highly enriched in differentiating OPCs. So you search for your gene of interest here, and you can colorize your gene of interest on the data set of OPCs. Another thing you might want to do is to compare different population of OPCs from this data set. So what you can do is select population 1 using this tool, and then you can select another population of cells and then click on this Venn diagram to sort of compare the difference between population 1 and population 2. So here, population 1 just happens to be the differentiating OPCs. Population 2 are one of the populations of quiescent OPCs. And here on the right, it shows you what are the different genes that are high in population 1 compared to population 2, and what are the genes that are high in population 2 compared to population 1. You can also colorize based on different marker genes of populations of your choice. And you can see here that some of the most canonical differentiating OPC myelinating oligogenes are represented here: CMP, BCAS1, EMPP6. So with that, I really want you guys to explore the data set. Let me know if you have any questions, and hope you guys find really interesting hidden pathways. So with that, I'd like to thank everyone in Dwight Burgle's lab at Johns Hopkins, especially Anya Kim, who's a graduate student in the lab who's been really wonderful and helpful. My collaborators, Loyal Goff, Ricky Kawaguchi, Peter Calabrese, Robin Franklin, and Bjorn Newman. And Bjorn is the person who performed the in vitro pharmacological assays to validate the single-cell RNA-seq hits. And then I'd like to thank my funding sources. So with that, I would be happy to take any questions in the chat or on Zoom. It can be either Korean or English. I will try to answer in Korean, but if I fail, I'll switch over to English and try my best. Thank you so much.